In this lab, we're going to teach you the two-step calibration method on the pH meters in lab. Then we're going to test some solutions. To begin this lab, you're going to rinse the storage solution off of your pH probe with deionized water. Then you're going to blot the deionized water off of your pH probe. After rinsing the pH probe, you need to set up the pH meter to two-step calibration. When the meter is ready, you're going to dip your pH probe into the pH 7 buffer solution, being careful not to hit the fragile probe on the sides of the beaker. Once the pH meter is reading and stabilized, you're going to press 7, then enter. Before testing the pH 4 buffer, rinse your probe with the deionized water into a waste container and blot the excess water with a CHEM wipe. After blotting the probe, place it in the pH 4 buffer. Wait for meter to stabilize, then press 4 and enter on the pH meter. The meter should be reading correctly at 4.0. While we watch the other lab mates calibrate the pH meter, let's talk more about what a buffer is.
A buffer is an aqueous solution that maintains the pH within a very narrow range. It will contain a weak acid and its conjugate base, or a weak base with its conjugate acids and their respective salts. Each buffer will have a certain working range, and in which they will be able to neutralize the solution. In order to do so, the buffer will donate either a hydroxide or a hydrogen ion and have a similar pKa value compared to the pH of the solution. An example of a buffer is bicarbonate in human blood.
Now that we know what buffers are and how they relate to this experiment, let's talk a little more about strong versus weak acids.
There are many types of acids, but the ones that we are focusing during this lab are Bronsted and Lowry acids. The definition of a Bronsted and Lowry acid is a species that is able to donate a proton. When looking at the difference between a strong and weak acid, it is important to note the ionization of the protons. In a strong acid, the protons will fully dissociate and the solution will not be able to reach equilibrium due to all of the reactants being used up. On the other hand, a weak acid is able to reach equilibrium due to the fact that it will not fully dissociate all of the protons and the reactant/products will stay at a constant concentration. In order to minimize the effect of this, we can use a buffer.
Now that everyone in our lab group has calibrated the pH meter, let's move on to testing some solutions. Our first is going to be apple juice. After dipping the probe into the apple juice and waiting for the meter to stabilize, we found the pH to be 4.13. Remember, in between each solution, you need to rinse off your pH probe and blot it with a CHEM wipe. The next solution we tested was orange juice. We found the pH to be 3.66. The third pH we tested was hydrochloric acid, also known as HCl. We found the pH to be 1.00. The last solution we tested was acetic acid. We found the pH to be 2.51. Remember to record all data in your lab notebook. We showed the solutions, the concentrations, and the pH to every figure the pH meter gave us.
When cleaning the pH probe, it is important to rinse off and dry the probe. Once the probe is clean, place it into the storage solution that came with the probe. Unplug the monitor from the wall and bring the probe and monitor back to the charging station. Unplug the probe itself from the monitor and slide it back into a spot on the charging dock. To finish the cleaning process, plug the monitor into the wall.